do some ring structure. I'll try to do a couple examples of those. So let's draw the Fisher projections. And I want to do, let's see, 1, 2, 6. Okay. Let's do OH, 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 OH. All right. So you should also, is there a question? Okay. You should all be able to name that structure from the, just as a sugar. So it's 6 carbons. So it's a hexose. And the OH on the highest number chiral carbon is on the right. So it's a D. And there's an aldehyde group on the top. So it's D-aldohexose. So on the exam, I tell you it's D-aldohexose with an OH group on numbers 2 to the right and 3 and 4 to the left. That's how it explains so you can draw that structure. And from this, you can draw the cyclical compound or the Haworth projection. Okay, so from the Fisher projection to the Haworth structure. All right, so we're going to have this oxygen right here is going to form back up and link with the carbon of the aldehyde group. And the hydrogen is going to attach. This hydrogen is going to attach to right here. And the oxygen is going to bond to that carbon right there. So what we're going to have is a 6-member ring with an oxygen in the ring. I'll make it look a little cooler here. I'm not very good at drawing these right on the computer. Okay. And you're going to have your bonds above and below the ring. Okay. And your CH2OH, your D sugars, CH2OH always goes on top. Okay. Now, the OH of this carbon here, at the number 1 position, if it goes up, there's 50% of the time to go up, 50% of the time it's going to go down. Okay. If it goes up, that's a beta. It's in the beta position. So beta D aldohexose. Okay. Now the other ones are going to be determined by the Fisher projection. So that's your basic structure right there. And then carbon number 2, let's put a purple one here. This is the second carbon right here. The OH group is on the right. So it goes below the ring. Okay. The next one is carbon number 3. The OH on the left, it goes above the ring. Okay. And the third one, the OH on the left, it goes above the ring. Okay. So the colors correspond to each of the carbons. If the OH is on the right, it goes down. On the left, it goes up. Okay. And so this is beta D aldohexose. Okay. If the OH group was down, this would be alpha D aldohexose. Okay. And that's how you determine this. Let's do another example. We'll do a 5-carbon one instead of a 6-carbon one. So let's put another aldehyde group up here. Okay. Put our OH, 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 then hydrogens. Okay, this is 5 carbons. So it's going to be a 5-carbon, a 5-member ring because oxygen is being one of them. Remember, this oxygen here, this oxygen right there, is going to become part of the ring. Okay? And the hydrogen next to it, this hydrogen right here, is going to bond to the oxygen of the carbonyl group. You get an OH group. That's the one that goes up or down. It turns out there's alpha or beta. That OH group with the hydrogen bonds. So we've got a 5-member ring now. I don't want to do that. Start it off right. Oxygen. So there's our 5-member ring. Okay. CH2OH. And our hydrogen. Okay. Where the OH goes up or down. Let's put it down this time. This is where the carbonyl group was. Okay. And then our number 2 oxygen is the number 2 carbons right here. And the OH is on the left, so it goes up. And our 3rd carbon number 3, the OH group's on the right, so it goes down. And that's it. And this is a alpha D, and it is a pentose. So alpha D aldo pentose, because the aldehyde group is on the top. That's how to draw a 6-member ring and a 5-member ring when you're doing them. So it's D2-deoxyribose. So it's a 5-carbon sugar. So it's a 5-carbon ring structure. So I'll draw the ring structure in the corner here. So again, it's 5 carbons with oxygen at the top. We draw a 5-membered ring. Okay. To the left, we have our CH2OH group. And our hydrogen, that's this right here. Okay. And this carbonyl carbon had that oxygen, the hydrogen added to it. So it's going to come up here, and let's put the OH down again, and then an H, and then going down, the 2nd carbon. Okay, 2nd carbon is right there. There is no OH group. So our 2nd carbon is 2 hydrogens. Our 3rd carbon, right here, the OH group is on the right-hand side. Right-hand side means it's below the ring. Okay. And there's D2-deoxyribose. That's the ring structure. Okay. That's how you would draw it. Okay. There it is here. This is our, okay, this is a beta because the OH group is up. Remember, this comes from this carbonyl group right here. Okay. Half the time it goes up, half the time it goes down. If it goes above the ring, it's a beta. If it goes below the ring, it's alpha. That's going to form our linkages. And then it's 2-deoxy, so the number 2 carbon, we're missing our oxygen, so it's just 2 hydrogens. The number 3, the OH group is on the right, so it goes down. And there's CH2OH. And that's the ring structure for D2-deoxyribose sugar. Okay. Okay. And this one is beta D2-deoxyribose. It's beta because this OH group is up. Like I said, 50% of the time it goes up, 50% of the time it goes down, because you're forming an OH group from this carbonyl group right here. So it's just going to be just random where it goes up or down. Okay. So disaccharides. Now you got 2. We've been drawing monosaccharides, 1-unit sugars. Disaccharide consists of 2 monosaccharides joined together through an oxygen bridge. So the oxygen bridges between the 2. And it bridges at a specific location. Okay. So in biological systems, monosaccharides exist in the cyclical form, and they're hemiacetyls or hemiketals with the OR or the OH group on them. Okay. When a hemiacetyl reacts with an alcohol, the product is a acetyl. Okay. When a hemiketal reacts with an alcohol, the product is a ketal. Just so we did before, exact same thing. So in the case of disaccharides, the alcohol comes from a 2nd monosaccharide because there's an oxygen OH group there. We get dehydration reaction, we lose H and OH, and it forms between them. Okay, so the acetyls or ketols, okay, are formed given the general name glycosides. So when they form the disaccharide bond between the two monosaccharides, they're called glycosides. And the carbon-oxygen bonds are called glycosidic bonds. Okay. So this is their acetyl group. And we have one R, one OH, and O on two alcohol groups attached. Okay. If this H was another R group, it would be a keto. Okay. So there's our acetyl group. Okay. And there's the two R groups with the ORs. That's a keto. Okay. The hemiacetyl is if one of these, if R was an H, and the hemiketal is if this R was an H. Okay, that's what we've gone over before. So that's your acetyl group, that's your keto groups. Okay, so a glycosidic bond is non-specific. It can occur between a hemiacetyl or a hemiketal and any of the hydroxyl groups on the second monosaccharide. Okay, so in biological systems, we commonly see only particular disaccharides like maltose, lactose, sucrose. Okay, we see those in the human body. Okay. These specific disaccharides are produced in cells because the reactions are catalyzed by enzymes. And enzymes are very specific in where they bond things to, because they bond by shape. Each enzyme catalyzes the synthesis of one specific disaccharide, one specific type. This ensures that one particular pair of hydroxyl groups on the reacting monosaccharides participate in the glycosidic bond. So it's either like a 1,4, 1,6, 1,2. It's a specific linkage that's going to form with carbon number 1 to carbon number 3, or carbon number 1 to carbon number 4. Okay, so it can bond specific because it's catalyzed by an enzyme, and enzymes are site specific. Okay, so our important disaccharides, the anomeric OH can react with another OH on an alcohol or a sugar. Remember all the OH groups on sugar or the OH group on alcohol? Those can react with another anomeric OH. Okay. This forms what we call a glycosidic bond. When you get a bond between two monosaccharides, it's called a glycosidic bond. Water is lost to form an acetyl group. Okay. It's a dehydration reaction. Remember, just like with alcohols. Okay, we dehydrated the bond, we took off one H and one OH, got water, then we have an oxygen bond between them. Okay. So here's our sugar on this side right here. Okay. This is an alcohol. Okay. The alcohol can bind by a dehydration reaction. So this carbon can combine with this oxygen, and we lose H and OH, and we end up with OCH3, which is right here. Okay. So we replace our hydrogen with a methyl group. That's how we bond the saccharide. This is a monosaccharide with an alcohol. Okay. And it forms an acetyl group. Because look at this point right here. Let's look at this carbon right here. That carbon, here's our blue carbon, has an OCH3. It has a hydrogen. Okay. It has an O and then an R group here with a mess going down there. And it's got another carbon R group attached here. Let me make that a little clearer. Okay. So what we've got, because this is what we're looking at here. There's a hydrogen attached, there's an OCH3, there's an OR, and then there's an R group. Okay. That is a acetyl group. Remember, you have OR, you have OR, you have your R group, and then we have a hydrogen. Okay. If that hydrogen was another R group, it'd be a keto. but that's an acetyl group right there, just like we've been dealing with before. So in a monosaccharide reacts with an alcohol, it forms water in a dehydration reaction plus an acetyl, okay, in the form of sugar, the same ring. Does everybody see that? Everybody see that? Sometimes it's hard to see what I'm looking at. That's why this carbon, so what's attached to this carbon here is a hydrogen, an OR group, another OR group, and another R group because it bonds to carbon. Okay, that's an acetyl. Do you have any questions on that? Because we're going to go on from there. So you got to be able to understand what that's looking like and how it's going to form when we form these other bonds. We all good? Someone give me a thumbs up or a yes or some. Perfect. You got it. All right. Well, let me open up the chat window too, just in case someone wants to chat. Sometimes chat. I'll bring that over here. And we can monitor the chat window. So maltose. Maltose is a type of disaccharide. Okay, it's formed by linking 2D-glucose at the 1,4 glycosidic linkage. So the number 1 carbon, the one that turns alpha or beta, remember, is going to link to the number 4 carbon of another glucose molecule. So it's going to link at the exact opposite side. Remember, 1 going down was a 2, then 3, then 4 going up, then 5, then your oxygen. Okay, maltose is malt sugar. It's formed as an intermediate in starch hydrolysis. How we form starch. Starch is a way to store glucose in the body. Okay, so reducing sugar is reducing sugar due to the hemi-acetyl hydroxyl group that's on there. Okay, that way it can be reduced. Can add hydrogen to it because it's a hemi-acetyl group. So this is your D-glucose. Remember, this is carbon number 1, 2, 3, 4, 5. And this is the oxygen from the chiral carbon with the highest number. And half the time that goes up, half the time it goes down. This is a beta linkage. Okay, so here's your oxygen that links them together. Okay, and again, this is number 1, number 2, number 3, and number 4, and number 5. So this is a 1,4 glycosidic bond linkage. Oops, I didn't mean to go that far again. Okay. So everybody see why it's 1,4. Okay. It links the number 1 carbon of one glucose molecule to the number 4 carbon of the other glucose molecule. Okay. This one is a alpha, and this one is an alpha because the OH group is down, and it was down on both of these. And you've lost water between them. Remember, there was an OH, 2 OH groups. So in other words, this had an OH, and then this one had an OH, and you lost water between them. That's not clear. Let me try to write a little clearer. And my writing's not great. But yet on one of them, you had an OH group. The other glucose, you had an OH group. And when they join together, you dehydrate it. You lost water between them. So you have an oxygen left that forms your glycosidic bond. And this has to be between, and the enzyme that's catalyzed this is very specific. It forms a 1,4 glycosidic linkage. Okay? That's where it's coming from. Okay, so formation of maltose. Okay, so it's alpha D-glucose and beta D-glucose. Okay. This is alpha because the OH group is down. This is beta because the OH group is up. So you're going to form a linkage between alpha D-glucose and beta D-glucose. Because remember, when it forms, it's a 50-50 mixture. When it forms a cyclical structure, which it forms in the body, that's what exists. Okay, so it's a dehydration reaction. We're going to lose water. We're going to take the OH here and the H here. And what we're going to be left with is this oxygen that bonds between them. and it's enzyme catalyzed. So it's a 1,4 glycosidic linkage or glycosidic bond. Okay. And this is beta maltose. It's not beta because of this. It's beta because right here. Okay. So once that bond forms, the two D-glucose molecules come together, one alpha, one beta, it forms maltose. And this one is beta because the OH group is up at the number 1 carbon. Okay, that's the number 1 carbon over there. Okay. Because this one's not going up anywhere. This is straight across. That's our oxygen bond between them. Okay, so everyone understands why this is called an alpha 1,4 glycosidic linkage, because both these OH groups here on this side were in the alpha position. Okay, the product is beta maltose. Lactose. We discovered maltose. Lactose is formed by joining beta D-galactose to alpha D-glucose to give a beta 1,4 glycoside or glycosidic bond or linkage. Okay. Lactose is milk sugar. That's a sugar found in milk. Milk is full of sugar. Parents do not realize that. If you have a newborn, okay, after 1 year, they should not have a bottle, period, because they don't brush their teeth. But if you put them in bed with a bottle, which you should not do, but if you do, never put them to bed with a bottle of milk. It will end up calling what's called grenade mouth. It will destroy every tooth in their mouth. It will turn rotten. All the teeth will rot out, and you can do what's called a baby denture, okay? Because of the lactose. Lactose is it's sugar. It will destroy teeth, okay? So if you put them in bed, and if you have a newborn, put in bed with a bottle, make sure it's just water. So lactose is your milk sugar. It's an energy source. It must be hydrolyzed to glucose and galactose for us to use it. We've got to break that bond. We have to break that 1,4 glycosidic bond to be able to use it as energy. Lactose intolerance results from lack of an enzyme to hydrolyze the glycosidic linkage of lactose. So if you can't break this linkage, this 1,4 bond, then lactose builds up in your system. You can't break it down to use it as energy, and it builds up. And that's what causes all the symptoms of lactose intolerance. Okay. So here's galactose, and here's glucose. Okay. And it's a 1. Again, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay. So the only difference between glucose and galactose is the position of the OH group on the number 4 carbon. In glucose, the OH group is down at carbon number 4. In galactose, the OH group is up or above the ring at carbon number 4. That's the only difference. Okay? And you're going to form a bond between the 1 and 4 carbons. And you're going to lose water. You're going to lose. Okay? So we're going to take this bond here. We're going to lose an OH and we're going to lose an H. And we're left with just an oxygen bond between them. And you get plus water. And it's going to be a 1,4 glycosidic linkage. It's 1,4, not 1,5, not 1,3, because it's enzyme catalyzed. It's not random. It's not these things floating around and then bonding to each other. There's an enzyme, and enzymes are site-specific, which means they line up both of them in exactly the same position every time. So the enzyme is going to grab one, grab the other, and it's going to force them together to remove water in the exact same position every time. Remember, enzymes are specific. Okay, so it's always going to be a 1,4 linkage for galactose and glucose to form lactose. Okay, that's why it's not random, because it's enzyme catalyzed. Okay, so galactose is identical to glucose, like we said, except at the number 4 carbon, the hydroxyl group is down on glucose and it's up on galactose. Okay, so the lactoglycosidic bond. Here's your beta D-galactose. Here's your beta D-glucose. Why are they beta? Because at the number 1 carbon of each one, the OH group is up. Okay, so they're both beta. Okay, the D comes from the Fischer projection, and we're going to lose water, OH and H, between there. So we're going to have an oxygen between the 1 and 4 position, which is right here. That's our beta 1,4 linkage. Okay, and it forms beta lactose. Now this beta does not come from here. Don't make that confusion. Okay, it comes from right here, because this OH at the number 1 carbon, okay, is up. If that were down, it'd be alpha. And like I said, it's a 50-50 chance with these as it's formed. So don't think that beta D-galactose and beta D-glucose will always produce beta lactose. It can produce alpha lactose. 50% of the time it's going to. Okay, so a lot of students, I'd say 80% on the exam are going to miss this, because they're going to say, oh, it's beta D-galactose and beta D-glucose. It's going to form beta lactose. And no, it can, but it can also form alpha lactose, because this is what determines alpha or beta. It's the position of the OH group at the number 1 carbon. If it's up, it's beta. If it's down, it's alpha. And that is not an enzyme catalyzed reaction. That's just a folding into the cyclical structure which exists at the pH conditions of the human body. Okay, it's just a natural folding of the ring structure to make it more energy, make it more stable. It's second law of thermodynamics. Okay, is that making sense?
Yes.
All right, perfect. All right. Okay, so there's your linkage, alpha 1, a beta 1,4 linkage. Okay, and it forms an oxygen down between galactose and glucose. So if you have lactose and you cannot break that bond, you cannot process this, and it will build up in your body. That's what causes the problems. You've got to have the enzyme that breaks that bond to be able to break it into glucose and galactose and able to metabolize it to form energy. You cannot utilize lactose. You've got to break it down to the monosaccharides to utilize it in the body. So lactose is the principal sugar milk of most mammals, which we talked about. Okay? To be used by the body as an energy source, you have to hydrolyze it. Okay? You have to add water back to it and break that bond apart to produce glucose and galactose. Okay, so glucose delivered by the hydrolysis of lactose is used directly in the energy reactions of glycolysis. We haven't gotten to glycolysis yet. We will in the next, I think chapter 18, we start glycolysis. Glycolysis means you're taking glucose, you're eventually breaking down to CO2 and water and relieving 30, 32, 33 ATPs, which is our energy currency in the body. All right. So a series of reactions is necessary to convert galactose into phosphorylated form of glucose that can be used in cellular metabolic reactions. So we use a glucose and we use a galactose, okay, to perform a reaction. You cannot use lactose. You have to break it down into separate monosaccharides. So in humans, genetic disease, galactosemia, is caused by absence of one of the more enzymes needed for this conversion. So in galactosemia, you cannot use galactose. Okay, so a toxic compound from galactose accumulates in people who suffer from galactosemia. Okay, so you're able to break the 1,4 linkage. You can use the glucose, but you cannot use the galactose. You can't phosphorylate it to put it back into the energy cycle. So this galactose builds up in patients with galactosemia. Okay, if not treated, galactosemia leads to severe mental retardation, cataracts, and early death. It just builds up the body. You can't do anything with it, and it's a toxin. Okay, so it affects your eyes, it affects your the brain, and then it leads to an early death. Okay, the effects of these can be avoided entirely by providing galactosemia infants with a diet that does not contain galactose. So you don't use lactose. You use soy milk. Use a different type of milk. Okay, you don't use lactose, because lactose breaks down into glucose and galactose. And galactose, if you have galactosemia, you're missing the enzyme that's going to convert galactose into a breakdownable product. And it builds up in the body that way. So such a diet, of course, cannot contain lactose, contain no milk or milk products. Okay? So patients with galactosemia, that's one of the reasons you cannot give them lactose. Because remember, lactose breaks into glucose and galactose. Okay, so galactosemia In order for lactose to be used as energy source, galactose must be converted into phosphorylated glucose molecule. Remember, glucose and galactose are real similar. They just have the OH in a different, the OH group and the carbon are in a different position. Okay. So when enzyme necessary for this conversion are absent, you have a genetic disease called galactosemia. Okay. People who lack the enzyme lactase, about 20% of the US population, are unable to digest lactose and have the condition lactose intolerance. So galactosemia and lactose intolerance are two different things. Galactosemia, you can break down lactose into glucose and galactose, but you cannot phosphorylate galactose to make it go into the Krebs cycle, into our cycle to break down for energy. You can't utilize it. So the galactose builds up and becomes toxic. People who are lactose intolerant, about 20% of the United States population, okay, cannot break the 1,4 glycosidic bond. So they have lactose, but you can't break it down, so it can't be utilized. Okay, so they're unable to digest lactose. They cannot break the 1,4 glycosidic bond between glucose and galactose. Okay, that's called lactose intolerance. For patients with lactose intolerance, undigested lactose remains in the intestinal tract. Okay, it can't be broken down. Causes cramps, diarrhea, and eventually leads to dehydration. Okay, lactose intolerant people, they eat ice cream, milk products, cheese, they don't do really well. Okay, some of the lactose is metabolized by intestinal bacteria. So some of your bacteria start to break it down because you can't. So some of the lactose is metabolized by intestinal bacteria and they release organic acids and carbon dioxide. What's carbon dioxide? Carbon dioxide is a gas. Gas builds up in your intestine. Not a comfortable feeling. Okay? You build up extreme amounts of gas inside your intestines. Lactose intolerance is unpleasant, but its effects can be avoided by a diet that excludes milk and milk products. So same thing. Same treatment, galactosemia and lactose intolerance have the same treatment. Just avoid lactose. Different reasons. Okay, galactosemia, you cannot utilize galactose. Lactose intolerance, you cannot break the bond, the 1,4 glycosidic linkage or bond between glucose and galactose. Okay, so the enzyme that hydrolyzes lactose is available in tablet form. So now, okay, when ingested with dairy products, it breaks down the lactose, preventing the symptoms. So lactose intolerant people can enjoy ice cream once in a while by taking this pill that contains the enzyme lactose, and it breaks the lactose down. But don't get the two confused with your patients. If you got a patient with galactosemia, you got a patient with lactose intolerance. They're treated completely differently. It's a totally different disease. Sucrose. Sucrose is the big sugar. That's the one that causes obesity, tooth decay, heart disease, all kinds of stuff in this society. Okay, that's table sugar, is sucrose. Sucrose is linked by linking alpha D glucose with beta D fructose to give a 1,2 glycosidic linkage. Okay, again, these are enzyme catalyzed reactions. So when these two things that come together, they're specifically put together by a specific enzyme that joins at a specific site. Sucrose will never be a 1,4. Lactose will never be a 1,2 link. Okay? It's enzyme catalyzed. It's not randomly coming together like the chemical reactions occur. Just like we've been doing all this time. You know, these chemical reactions, they hit, they form reactions, they form products. Okay? Doesn't happen that way. An enzyme grabs fructose, enzyme grabs glucose, they line up in specific one-way pattern, and they form that linkage. Okay? So these are enzyme catalyzed reactions. So sucrose is formed at alpha D glucose, beta D fructose to give a 1,2 glycosidic linkage. Okay, it's non-reducing, so don't have a reaction in the Benedict's test. It doesn't reduce, so it doesn't cause oxidation of something else like a reducing sugar would, like a monosaccharide. So the glycosidic oxygen is part of acetyl and a ketyl. Sucrose is an important plant carbohydrate. Okay. Sucrose is water soluble. Most sugars are water soluble. Okay. They have a lot of OH groups, a lot of polar groups on them. Okay. They're easily transported in the plant's circulatory system. Okay. The phloem and the xylem, the tubules in the plant, they're water soluble. They flow in that really easily. Okay. Cannot be synthesized by animals. Okay. We cannot make sucrose. Animals cannot synthesize sucrose. Sucrose is called table sugar. That's what you put in your tea. That's what you put in your cereal, whatever, your coffee, whatever you want to put in it. That is sucrose. It's also called cane sugar. It's also called beet sugar. Okay? And it's linked to dental caries. That's what causes Streptococcus mutans in your mouth. That's uses sucrose as a metabolite, and it produces acid as a byproduct, and that's what dissolves your teeth. Okay. So high concentration of sucrose produce a high osmotic pressure, okay, which inhibits the growth of microorganisms. So we use it as a preservative. Okay. We get high concentration of sugar. Okay. It creates a high osmotic pressure because it's polar and it draws water molecules towards it. So it produces a large osmotic pressure. It's widely used as a sweetener. That's what we use it for. It's estimated the average American consumes 100 to 125 pounds of sucrose every year. And just think about that for a second. Take a tablespoon of sugar and then put it on a scale and another one on a scale. Think about how much it takes to get to 125 pounds of that. And that's what the average American consumes in a year. That's a tremendous amount of sucrose. Right? It's been suggested that sucrose in the diet is undesirable because it represents a source of empty calories. Okay? Contains no vitamins, no minerals, and nothing that's good for you. It just tastes better. That's what it does. Makes things sweeter. Okay? The glycosidic bond that's formed in sucrose, like you said, is a 1,2 glycosidic bond. Okay? So this is alpha glucose, alpha D-glucose, and beta D-fructose. Again, it's alpha because this OH group is down, beta because this OH group is up. And you're going to remove water between the two, leaving this oxygen right here to form your glycosidic bond. Okay? Now it's going to form, now remember fructose is a ketose. It's a ketone sugar. It doesn't have an aldehyde on top. Glucose was an aldose, so it had a 1 CH2OH group. Okay. Fructose has 2. Okay, because there's a ketone on top. Remember, our sugars had the ones you've been dealing with have HCOO, CH2OH. Okay. Fructose, if we did a Fischer rejection, was CH2OH, carbon, CH2OH. Okay, so there's this is an aldose, this is a ketose. So you have 2 CH2OH groups, one here and one here. Okay, so this is the number 2 carbon on fructose. This is the number 1 carbon on glucose. Be careful to make that mistake because the number 1 carbon is here in the CH2OH group, because you always count as it goes across. Just because it's not part of the ring doesn't mean it's not your first carbon. It's the top carbon. It's this carbon right here is number 1 in fructose. Okay, so it looks like it's a 1-1 linkage. You're just looking at just casually, but think about it. It's a 1-2 linkage. It links the number 1 carbon of glucose to the number 2 carbon of fructose. Now the number 2 carbon is in the same position as the number 1 carbon of glucose because it's a ketose, not an aldose. Remember, glucose is an aldose, fructose is a ketose. That making sense? I know it's a strange concept. It's easy to miss if you're not careful. But you gotta remember, this is the number 1 carbon right here for fructose. So this is a 1-2 linkage. It goes alpha 1-2 to alpha 1 to beta 2. Because again, this was beta. This is alpha. So it's an alpha 1 beta 2 linkage. So far, so good. So we got the number 1 carbon here, attached to number 2 carbon here. Okay. And again, it's an alpha 1 beta 2 linkage, and then we get rid of water between them. So it's a dehydration reaction, just like we always done. And remember, these are enzyme catalyzers, site specific. So sucrose is always going to be a 1-2 linkage. Does that make sense? Let's see who's on there. Yeah, we're all good. All right, perfect. So blood transfusions and these blood group antigens, okay? These are our sugar groups. So these sugars we're talking about have important properties in the human body. These determine our blood group antigens, okay? So the first blood transfusions were tried in the 17th century. This used animal blood to replace human blood. Okay, that was lost by hemorrhages. Okay? Many people died of this, okay? Animal blood and human blood don't mix. Transfusions were banned in much of Europe because of it. Okay? Transfusions from human donors were somewhat less lethal, but violent reactions often led to death of the recipient. Okay? By the 19th century, transfusions had been abandoned as a medical failure. Once you had to learn what these sugars were that are on the red blood cells. In 1904, Langston performed a series of experiments on the blood of workers in his laboratory. Okay, he took blood samples from his coworkers. He separated the blood cells from the serum, okay, the liquid component of the blood, and mixed these samples in a test tube. So your plasma is a liquid portion of your blood. Okay, he mixed the serum from one individual with blood cells from another individual. Then he observed that some of the serums caused clumping, okay, and what's called agglutination of the red blood cells, which means he mixed some of the serum together and they clumped together, forming a precipitate. Okay, that would clog the bloodstream, that would cause death. Some of them mixed together and it didn't do it. See, I figure out why. So the agglutination reaction always indicated that two blood types were incompatible. So your blood types are incompatible, you get agglutination. And the transformation could lead to life-threatening reactions because it clogs up, it stops the red blood, it stops your blood from flowing. It clogs up the entire artery. Okay. As a result, he showed there are 4 human blood groups. There's A, there's B, there's AB, and there's O. Okay. These are sugar molecules we've been talking about that are on the outside of the red blood cell. Okay. There's a type that's like an A. There's a type that's a B. There's a type of blood cell that's A and B on it, and then O has neither A or B. So the 4 types, you got 4. In fact, I'll even use red for red blood cells. You got 4 types of red blood cells. On these, there's these little A sugars. On this one, there's B sugars. Okay. On this one, there's A and B sugars. And on this one, there's no sugars on it at all. Okay. If you mix the wrong type, they agglutinate, they form clumps, and it's fatal to your patient. Okay. So here's your group A, group B, group A and B, and group O. Okay. Now you have antibodies in your plasma. These bind to the red blood cells and cause this thing to clump together. So if you have blood type A, you have anti-B in your body. So if B is introduced, these bind to cells and cause agglutination. If you're type B, okay, you have antibody A that will bind to the A blood cells, okay, and cause agglutination. If you group AB, you don't have any antibodies because you have A and B on your own blood cells. Okay. If you group O, you have nothing, so you have A and B antibodies in your blood. So if you give a person that has type O blood, either A or B blood, these antibodies are going to bind to it and clump it. Okay. If you take a person with AB blood, okay, you can give them any of those, and not going to bind to it. So antigens on your red blood cell, these are A antigens, B antigens, A and B antigens, and you have no antigens in group O. So the difference of these blood groups, we've had differences in the oligosaccharides. Oligosaccharides are the sugars that are attached to proteins and lipids of the red blood cell membrane. Okay. They all have a common core consisting of beta D N acetylgalactosamine and galactose, N-acetylneuronic acid, which is sialic acid, and L-fucose. Okay. Those are oligosaccharides. It's the terminal monosaccharide of oligosaccharide that distinguishes the cell and governs the compatibility of the blood type. It's the end of that determines whether it's A or B, what's sticking out of the red blood cells. So these are your red blood cells. These are the antibodies. If these antibodies react, okay, if this is antibody B and this is a B antigen, then they react and they form these complexes and they stick everything together. That's what causes the problem. Okay. So, but if this is a, say if this was an A antigen and you had anti-B in there, you would do just fine because it's not going to find it as foreign. It's not going to mark it. Okay. Anti-B looks for the B antibodies to bind to. Okay. So the A blood group, okay, has antigen beta, D, and acetylgalactosamine at its end. The B blood group has antigen alpha D galactose at its end. So it's different sugars. Okay. Type O don't have any sugars on the blood surface. Only the core oligosaccharide is present. So there's no end marker on it to identify it as A or B. So some of the attacks on type AB blood have the different types of terminals, beta D N, acetylgalactosamine, whereas others have the alpha D galactose. So A has a specific type, B has a specific type, O has none, and AB has both arranged on the blood cell. So why does agglutination occur? Like we said, the clump reaction occurs when incompatible blood types are mixed. So you have an antigen antibody reaction. Okay, antigens are large molecules, okay, often portions of bacteria or viruses that stimulate the immune defense of the body to produce protective antibodies. That's how serums work. That's how vaccines work. The COVID vaccine. Okay, that's how it works. Okay, it produces the COVID response in the body. The body recognizes the outer markers, the sugars on it. We build antibodies to that, and we give you a subclinical dose so you don't get the disease. Okay, that's how the flu vaccine works. Okay, the antibodies are made in response to this, and it kills off the sub-infection that you're given. Now the antibodies are made in the body, and they can respond when you're exposed to the new virus, and they kill it before it has a chance to replicate in your body. That's how the COVID vaccine works. Okay. That's why you get sick with the COVID vaccine, okay? Because it's giving you a subclinical case of it, making your body form antibodies to it, and that when exposed to it, you can react to it. Now, for your flu vaccines, okay, that's not the case. Flu vaccines are an attenuated virus. The virus is dead. Okay, there is no way you can get the flu from the flu vaccine, period. Okay, it's killed. All that's being exposed to is the outer shell, so your body makes antibodies to that. And that way you're inoculated with it. Now you can still get the flu, don't get me wrong, because there's so many different cases of the flu, so many variants of it. The CDC comes out with, they pick 20 every year, and they inoculate for those 20 common viruses. If you get the 21st one, you're still gonna get the flu. But you have a much better chance of not getting with the flu vaccine. So antibodies bind to the foreign antigens, and they help destroy them. Okay, so people with A blood also have antibodies against type B blood. They have anti-B in their serum, which is a liquid part of the blood, the plasma. And why is this not working? So if there we go. If the person with type A blood receives a transfusion of type B blood, the anti-B antibodies, okay, react with the B cell, with the B sugars on the red blood cell and cause it to clump together because it forms this antibody antigen complex. Okay, and so that can result in death because it blocks the artery where it's at. The blood all clumps together. Individual type B also produce anti-A antibodies, so they cannot receive a transfusion from type A individual. Okay, so those with AB blood don't have either A or B antibodies. If it did, it would destroy their own cells. So they don't have that. Okay, type O has no A or B antigens on their blood cells, but they both have anti-A or anti-B antibodies. Okay, so AB would be the universal recipient. They can take blood from anybody. Okay. Type O is a universal donor. You can give blood to anybody. Okay, because if you have AB blood, okay, you have no antibodies in your body, in your bloodstream to bind. So you can take blood from an A take blood from B, take blood from an AB, or take blood from his O patient. Okay. Now there's a little more to it. We get a little deeper from that, but because there's RH factors or one of those. And so there's other things involved. This is a simplistic view of it. This is an overview of it. Okay. Type O blood, you have no antigens, so you have anti-A and anti-B antibodies. So if you take type O blood, you can type A, they're going to die. Type B, they're going to die. Type AB, they're going to die. All they can take is type O. But they can give blood to anybody because they have no markers on their blood. So if you have anti-A or anti-B antibodies in your serum, the type O has nothing to bind them to. So you're not going to get agglutination. So type AB is your universal recipient. Type O is your universal donor. That's why there's a big push right now with the American Red Cross for type O blood. They've been running out with all the things that have been going on the crazy year. But the shelves are almost empty of type O blood because that's the universal donor. So there's big advertisements for people with type O blood to give blood right now. Because it presents both types of antibodies, type O individuals can receive transfusion only from people with type O blood. They can't get from anybody else. Okay. AB individuals are universal acceptors. Okay. O individuals are your universal donors. So AB can receive blood from anybody. Type O individuals can give blood to anybody because of the sugars we talked about on the surface of the red blood cells. Poly means many, saccharides means sugar. So now we're going to deal with, we've had monosaccharides, we had disaccharides. Now we're going to put together polysaccharides, which is a whole bunch of them. And we have starch. Starch is a linkage of monosaccharides. Starches are the storage form of glucose that is found in plants. So when you produce glucose, either by ingesting it in or by photosynthesis in plants in this case, okay, when you use it for energy. When you have extra, you have to store it for energy later on. Okay, plants store glucose in the form of starches. Okay, those are polymers of alpha-linked glucose. If the links are 1,4 linkage, the polymer is linear. Okay? So if it's a series of 1,4 links, 1,4, 1,4, 1,4. Remember, 1,4, they're linking at both ends. So if it's, remember, glucose is okay. This is number 1. This is number 4. So if you're linking these, they're going to form a straight line. Okay, they're not they're not offset. So if it's only 1,4 linkages, the polymer is going to be linear. If it's linear, it's called amylose. That's a linear starch, all 1,4 linkages. Amylose usually assumes a helical configuration, about 6 glucoses per town, because there's hydrogen bonding. Remember all these glucose units have OH, okay? Have hydroxyl groups. Okay? Hydroxyl groups form hydrogen bonding, so it's going to link and build upon itself. So it's going to start to form a helical structure, just naturally through hydrogen bonding. And it's going to be about 6 glucose for every turn of that. Okay? It makes about 80% of plant starch. So 80% of plant starch is linear amylose 1,4 glycosidic linkages. Okay? If you have 1,4 and 1,6 linkages, so remember the number 6 carbon is right here. That's that top carbon. And it's going to bind to the number 1 position. That's going to put a bend in your straight line. It's not going to be a straight line anymore. So if you have 1,4 and 1,6, then the structure is branched and it's called amylopectin. So if it's all 1,4 and linear, it's called amylose. These are starches in plants. Okay, 1,4 linkages, all linear, it's amylose. 1,4 and 1,6 linkage means you can get a straight line and a branch, straight line, then a branch. So you can get branches on it. Okay, that's amylopectin. So you have to remember it's amylopectin and amylose. So amylose is linear, amylopectin is branched. Okay, highly branched, it branches approximately 20 to 25 glucose units per branch. Okay, so plants have the ability to use the energy of sunlight to produce monosaccharides like glucose. Okay, from carbon dioxide and water. So plants take carbon dioxide and water, and produce glucose and oxygen. We take glucose and oxygen and produce carbon dioxide and water to make energy. So plants use photosynthesis to produce monosaccharide glucose and then from carbon dioxide and water. Okay. Although sucrose is a major transport form of sugar in plants, okay, a starch is the storage form in most plants. So sucrose is transported through the plant as it goes from one cell to another to break down and provide glucose, provide energy. Okay. But the storage form in plants is starch. That's your polysaccharide, amylose or amylopectin. Okay, so these plants store glucose in what's called starch granules. Okay, all plant cells contain some starch granules, but in some seeds like corn, as much as 80% of the cell's dry weight is starch. Okay, so that's why you have high fructose corn syrup. Okay, 80% of corn, of its dry weight, is starch, is glucose molecules, is sugar. Okay. Starch is a heterogeneous material composed of glucose polymers, of amylose and amylopectin. Remember, amylose is your 1,4 linkage. It's linear. Amylopectin is 1,4 and 1,6 linkages, and it's branched. 80% is going to be amylose. Other 20% is going to be amylopectin. Okay, so amylose. What's this? Accounts for 20% of the starch of a plant, is a linear polymer alpha D glucose molecules connected by C1 and C4 of the second glucose. And amylose accounts for 80% of the starch of a plant. And that's a C1, C4 alpha. It's a 1,4 linkage between the two glucose molecules. A single chain connects with 4,000 glucose units. So 4,000 glucose can bond to the unit, and then it coils around itself to form a helical structure. So amylose coils up in a helix, repeats about every turn, has roughly 6 glucose units. So it can connect to 4,000 glucose units, which is a long linear chain. Okay. So this is what amylose looks like. Amylose is your alpha 1,4 linkage. Again, it's alpha because each one's facing down. Okay. And it's bonded at the number 1 position here and the number 4. Number 1, number 4. And it's a 1,4 linkage. Okay. And it links in a linear fashion because it's a hydration reaction. You lose water between each one. And once you have this long chain, it gets to 4,000 long, then it starts to bend upon itself. Okay, and it forms a helix. That's because of the hydrogen bonding between the hydroxyl groups. Okay, it naturally starts to fold into a helical chain. So amylose, remember, is linear, and it's 1,4 glycosidic linkage. Okay, amylose is degraded by 2 types of enzymes. Okay, it does no good to store it if you can't break it down when you need it. So the plants store it so they can have that energy when they need it. Okay, and you can break it down with 2 types of enzymes. They're produced in the amylose in the human body, where we eat starch. Produced in the pancreas, which is secreted in the small intestine. So the pancreas produces, that produces insulin, glucagon. It also produces enzymes for digestion. Okay, so enzymes produced in the pancreas, which are secreted into the small intestine, and the salivary glands, which are secreted into the saliva. Okay, so alpha amylase cleaves the glycosidic bonds of amylose chains at random along the chain, producing shorter polysaccharide chains. Okay, then beta amylase, okay, cleaves each individual disaccharide maltose from the reducing end of the amylose chain. Okay, so then it breaks it down into further and further. So the alpha amylose just randomly chops it up. The beta amylase comes in and shaves off every disaccharide. Okay, so it cleaves off every 2 of them. It forms a consistent pattern there. So the maltose is hydrolyzed into glucose by the enzyme maltase. Okay, so how amylose is broken down, remember? So amylose is your linear storage, right? Okay, and it's this long chain of glucose molecules. So you have two amylases. You have alpha amylase, which kind of just chops it randomly and cuts it into more manageable pieces. Then you have beta amylase, which comes in and breaks off every two sections. Okay, so every two monosaccharides, so it forms a disaccharide maltose. Then once you have these 12 dissection maltose, you have the enzyme maltase, and that breaks the two maltoses apart into two glucose molecules. And that's how we can eat starch and utilize it as energy. Okay, so that's how we break it down. And the enzymes are produced in the pancreas and in the saliva from your salivary glands. Okay, the glucose is quickly absorbed by intestinal cells and used in the cells of the body as a source of energy. Okay, amylopectin. Remember, amylopectin is the branched one. Okay, it's highly branched amylose, which the branches are attached to the C6 and C1 glycosidic linkages. There's a should be an arrow there. So alpha 1,6 glycosidic bonds. Okay, the main chain is 1,4. That would be amylose. Amylopectin is the alpha 1,4 and alpha 1,6. You have two types of linkages now, 1,4 and 1,6. The 1,6 linkage causes the branch. 1,4 linkages are linear. Okay, each branch contains up to 20 to 25 glucose units, and there are so many branches the main chain can scarcely be distinguished. The branches all over the place. Okay, so we compare amylose to amylopectin. This is amylose. This is a 1,4 linkage. Okay, and it's linear. So amylose is linear, all alpha 1,4 glycosidic bonds. Amylopectin. Okay, again, this is 1,4, 1,4. Now you get a branch. This is the number 6 carbon. This is the number 1 carbon. Okay, so now you get a 1,6 linkage. Okay, so you have alpha 1,4 linkages for amylose with a straight bond. The alpha 1,6 puts a bend in it, and it forms what's called amylopectin. So the 1,4 linkage is a linear line. When you put a 1,6, it kicks it up and forms a branch. And then you go back to your 1,4 linkages to form another line across to about 20, 25. Then it'll branch again. But every 20, 25 glucose molecules, it'll branch again. Okay. So what chemical reactions are catalyzed by alpha amylase and beta amylase? Alpha amylase does what? It takes amylose and just chops it into pieces. So the alpha amylase just chops it into pieces. It's a big long chain of glucose molecules, and it just randomly comes in and chops it up. So now you have shorter sections to deal with. Then the beta amylase comes along and breaks off every 2 disaccharides for maltose. It just cuts everything into 2 units. And now you have just these disaccharide units. Okay, that's what alpha amylase and beta amylase do. Then maltase comes in and breaks down those 2 units into glucose. Okay, so it says alpha amylase catalyzed reaction that breaks the glycosidic bond of amylose chains at random spots along that chain, the amylose chain. This produces shorter polysaccharide chains that are acted upon by beta amylase. Beta amylase breaks the bonds into disaccharides, means 2, which is 2 glucose form maltose. Okay. And then maltase comes in and breaks down the maltose. So that's how amylose is broken down by alpha amylase, beta amylase to form maltase, and then maltose forms maltose. I'm sorry, the maltase comes in and breaks that one down. So you talked about starch, storage of glucose in plants as amylose and amylopectin. Amylose is 1,4 glycosidic linkages, is linear. Amylopectin is 1,4 and 1,6 linkages. The 1,6 causes a bend and causes branches. Glycogen. Glycogen is a major storage of glucose molecules in animals. Okay. Same thing as starch in plants. We have glycogen in animals. again, when you eat, the extra energy you take in that you don't use has to be stored for later on. And we store it as glycogen. Okay, it's a highly branched chain, just like amylopectin. Okay, has more frequent branching though. Remember, amylopectin branched every 20 to 25 carbons. Glycogen branches about every 10, not carbons, 20 to 25 monosaccharides or glucose units. Glycogen branches every 10 glucoses. Okay. Glycogen is stored in the liver. Okay. It is stored in muscle cells. Okay, so it's stored in liver and muscle cells. Glycogen is a major glucose storage molecule in animals, like we said. Structure is similar to amylopectin. The main link is 1,4 glycosidic bonds, and then has numerous 1,6 glycosidic bonds also. There should be an arrow there, 1,4 and 1,6 glycosidic bonds. So the 1,6 glycosidic bonds provide the branching points of the chain. Glycogen differs from amylopectin, only having more and shorter branches. Okay, so it's the exact same thing as amylopectin, except it has more branches because the units are smaller. Instead of 20 to 25 monomers, it has about 10 monomers. So it has more branches and there's shorter branches. That's the only difference between glycogen and amylopectin. So glycogen and amylopectin, this is amylopectin here. You can see how it has branches, runs about 20, 25 units. And this is glycogen, about every 10 units, it forms another branch. Okay, but they're both the same. They both have alpha 1,4 glycosidic linkages that form a linear structure, and then 1,6 alpha 1,6 linkages that form the branch in the structure. Okay. So amylopectin, glycogen are very similar. Cellulose. Cellulose is a major structural polymer in plants. It's a linear homopolymer, so you can compose all of beta-D-glucose units, okay? And they're linked at beta 1,4, not alpha 1,4. So again, they're above the chain when the linkage occurs. Okay, so cellulose is a beta 1,4 linkage as opposed to an alpha 1,4 linkage of amylose or amylopectin or glycogen. Okay, repeating disaccharide of cellulose is B-cellulobiose. That's the disaccharide of it. Animals lack the enzyme necessary to hydrolyze cellulose. Okay, so we cannot break down cellulose. Okay, we don't have the enzyme to break the beta 1,4 linkage. We can break the alpha 1,4 linkage, but we cannot break the beta 1,4 linkage. So the bacteria in ruminants like cows can digest the cellulose, so they can eat grass, etc. So they can digest the cellulose because the bacteria in their gut is what's breaking it down. They're not breaking it down. Animals don't have the enzyme to do it, but the bacteria that live like in the cow's stomach can break it down, and therefore the cow can utilize that energy. They can break that beta 1,4 linkage. So cellulose is a beta 1,4 glycosidic linkage. See how it steps up? So it's the 1,4, okay, and it's beta up on top and forms that linkage there. Okay, so that's your beta 1,4 glycosidic bond to form cellulose. So the most abundant polysaccharide, okay, indeed the most abundant organic molecule in the world is cellulose. Okay. It's beta D-glucose units linked by a beta 1,4 glycosidic bond. That is the most common polysaccharide in the world, is cellulose. Okay. A molecule of cellulose can contain about 3,000 glucose units. Okay. But the largest ones produced by Alga valonia contains 26,000 glucose molecules. Okay, so they can be huge. Okay. That is the major storage form in plants. Okay, cellulose is a structural component of the plant cell wall. Remember, plant cells have a cell wall, human cells do not. In contrast to glycogen, amylose and amylopectin, cellulose cannot be digested by humans. We cannot break it down. We don't have the same bacteria in our guts as cows do that break down the cellulose. We cannot digest cellulose. Okay. The reason is that we cannot synthesize the enzyme cellulase, which hydrolyzes, adds water to that beta-1,4 glycosidic linkage of the cellulose polymer. Okay, so we cannot break the beta-1,4 glycosidic linkage. So when we eat cellulose, it goes right through our bodies. It does not get broken down. It does not get digested. Okay. Only a few animals like termites, cows, goats are able to digest cellulose. Okay. And cows and goats can do it only because the bacteria in their gut are breaking it down. They're not producing that enzyme either. But the bacteria that live in their intestines, their gut, allow them to utilize that as a food source. So these animals have within digestive tracts microorganisms that produce the enzyme cellulase. Okay. The sugars released by the microbial digestion can then be absorbed and used by these animals. So the animals, termites, cows, goats, okay, these microorganisms in their digestive tracts produce cellulase. The cellulase then can break down cellulose into glucose units, and those glucose individual units, those monosaccharides, can be used as energy by the animals. In humans, cellulose from fruits and vegetables serve as fiber in our diet. Okay? That's our fiber content. Because we can't break it down, it serves as fiber for our diet. So what is the function of cellulose in the human diet? Okay? It's fiber. Okay? Cellulose functions as fiber in the human diet since we can't digest it. We cannot break down that beta-1,4 glycosidic linkage. Okay? The structure of cells that requires the beta enzyme cellulase, okay, to break the beta-1,4 glycosidic linkage of the polymer. And we cannot synthesize the enzyme cellulase. So here's cellulose. You can see its steps going up each one. It's still the 1,4 linkage, but it's a beta linkage now. It doesn't come down to form a linkage straight across like linear, but it steps up as an angle. And to break this bond, we have to add water to this bond. So you end up with OH here and OH here, and cellulase breaks that bond and puts water back in between those molecules and allows you to break it, or you hydrolyze it.